proteins. And these proteins are important because they control all aspects of the cell. So any mutation in one portion of this process can lead to severe defects. And my lab mainly focuses on what happens in messenger RNA, because any defects here can also affect the proteins that are being manufactured. So we looked at what happens when pre-mRNA is processed into mature RNA, because there's a series of steps, like adding a FICAP methane binding cap and polyadenylation, and finally exporting to the cytoplasm. So all along this route, there are proteins that attach to the transcript and allow and guide the transcripts to be exported out to the cell where they can be successfully translated. And the protein that I'm working with is called ZC3H14, which is located on the poly tail right here, which is basically a string of adenosine residues on the 3' region of the transcript. And this is associated with controlling the poly length and making sure that it doesn't degrade in the cytoplasm in the nucleus. So this gene has a unique domain, ZCCH zinc finger domain, and this allows it to bind to the polyase strand. And we've seen that it localizes to nuclear speckles and interacts with the splicing factor. So we think that it does play a factor in mRNA processing. And understanding the mutations and what happens is important, because the change in proteins can lead to something like mental retardation, or what I'm focusing on is specific types of cancer, like clear cell renal carcinoma and some breast cancer types. And one of these breast cancers is looking at ER positive or ER negative breast cancers. ER is basically an estrogen receptor, which acts as transcription factors, and it's regulated by the steroid hormone estrogen, and it leads to tumor growth, so that's important to study. And the status is used to determine prognoses of patients and see what treatment to give. So it's important to understand how it's regulated in the cell. And as you can see, across the 7 studies, ZC3H14 expression is higher in the ER positive tumors. So we wanted to look at what regulates what. It could either be that ER regulates ZC3H14 or vice versa, because these studies do show a correlation. So to test the first theory, induction with estrogen does not show a rise in ZC3H14 expression, so that probably isn't the case. And for the second theory, to test that, we knocked down ZC3H14 using siRNA, and that does show a concomitant decrease in ER mRNA levels. So we think that this is probably the method by which it works. But to understand it further, we have to understand how these RNA binding proteins, how would they work, and by what mechanism do they recognize where to bind. And so to do that, we looked at the yeast ortholog, NAB2, to study what happens in ZC3H14, because they're similar genes. As you can see, that they have, they're highly conserved, which means that the regions like the N-terminal, which is used for exporting to the cytoplasm, and the glutamine-rich regions, and the nuclear signaling regions